started. So welcome everyone to this week edition of Extracellular Vesicle Club, the weekly events of International Society for Extracellular Vesicles. My name is Olesia and I'm a research associate in the lab of Dr. Ken Witwer, and it's my pleasure today to introduce Gijung Kwak, who's gonna present a paper which is just recently published in Journal of Extracellular Vesicles. And today we're gonna talk about using extracellular vesicles in therapeutics. Okay, feel free to share your screen and welcome. Just a reminder for everyone will be muted until the end of the presentation, and after presentation is done, I will allow people to unmute themselves and ask questions. If you have questions during the presentation, please put them in the chat below.
I'm Gijung Kwak, who working in the Center for Nanomedicine at Johns Hopkins University. Today, I'm here to present the extracellular vesicle-associated adeno-associated viruses for inhaled gene delivery. Many of the lung gene therapy is the very focused in the gene delivery field. There is a couple of monogenic mutations on the lung disease, including cystic fibrosis and also the ARPA trypsin deficiency. So many kind of gene delivery strategies are targeting for the lungs for more than 30 years, but there is no clinical options for now. All failed to deliver the enough genes on the airway cells on the lungs. Recently, there is a major hurdles to inhibit the gene delivery efficiency to the lungs is discovered as the mucus layer and also preciliary layer. Mucus is the polymerized mucin compositions characterized with the hydrophobicity and also sticky properties. So many of inhaled materials can be trapped in the mucus, cannot be penetrate to the deep side of the lungs. And there is a clearance process in the lungs, the mucociliary clearance. So trapped materials can be cleared from the mucus. But there is many kind of evidence are revealed with the EVs. Many of lung disease, and then even for the healthy lung, there is many of reports that EVs can exist in the mucus. In the diseased lungs, they can make some important roles for the signal transportations between the cell to cell, even in the mucus. So we try to focus on the EV as a vesicle to deliver the drugs. One of the recent publications showed that the milk exosome can penetrate the porcine intestinal mucus, and then they can deliver their model drugs through the mucus penetration. So we searched the loading method into the EVs. There is multiple papers are reported about the EV associated adeno-associated viruses, EV AAV. The AAV production with the cells usually conducted with the multiple AAV producing gene transfections. Usually hexel is used for the productions. When the AAV produced from the cells in their egrazing steps, they can be utilized at the EV secretion pathway. So AAV also can secrete it with EVs. So they can be loaded inside of the EVs or associated, attached to the outside. So they have some advantages about the increase its intracellular uptakes and then antibody shielding. One of the major challenge of the AAV is the anti-AAV antibodies. And then there's multiple engineering options we can apply it to the EV-AAV. We utilize the AAV6, AAV serotype 6 here because they are known to be less adhesive to the mucus in the previous paper. So our hypothesis was EVAV6 is less mucoadhesive. Probably their association with the EVs can enhance the mucus penetrations. If AAV associated AAV6 does not compromise the mucus penetrating property on the EVs. Firstly, we characterized EVAV6 first. The first Western blot data showed that EVAV6 showed viral capsid protein BP123, and then extracellular vesicle markers. The synthetin-1 is the intracellular EV marker, and then CD9 is tetraspanin EV membrane marker. All proteins can be detected with the EVAV6 samples. And then the second is the ExoView analysis. We utilized the original normal EV and then EVAV6 samples for the analysis of the EV populations. They showed almost similar population patterns based on the EV specific markers, synthetin-1, multiple tetraspanins. Then we measured the size of the materials. The AV6 showed almost 25 nanometer size. That is original size of the AAVs. And EVs was measured in about 100 nanometer size. And EVAV6 showed 120 nanometer sizes. Interestingly, the size difference is about 20 nanometer. That is correlated with the AAV size. And then the bottom images is the TEM images, transmission electron microscopy images. Each AAV6 and EV images are showing their own morphology. And EVAV6 images show that association of the AAVs and EVs, internalize it or externalize it. And we also prepare the post-mixture sample, separately prepared EVs and separately prepared AAVs, and then we mix them together after the separate preparations, and then incubate in the room temperature for 30 minutes. With that conditions, in the TM image, there is almost no associations. In the simple room temperature, the associations was not conducted. Then we also curious about the stability of these associations. We applied the sonication and nebulizations is kind of physical stimulations to the EVAV6. The sonication condition is very have a strong intensity. This condition can be utilized for the 40 nanometer nanoparticle encapsulations into the EVs. So it was very strong, but after that sonications, the EVAV6 size was maintained well. And then after the nebulization, also the size was well maintained. So the associations can be tolerable for the physical stimulation here. And we can measure the diffusion rates in the CF mucus from obtained from CF patients. The upper two videos is our other types of nanoparticle that can be highly pegylated, so they can be freely moved in the PBS and serum. The upper figures is the mean scalar displacements. That is, we can image these videos. The time interval is 67 milliseconds. Then we can measure each location displacements based on this image, and then we can calculate the distance, average distance in 1 second's time scale. So with this analysis, the mean display, like MSD value, is proportional to diffusion rates. So upper two videos show that our particle can be diffused well in the PBS and serum. We also did the same method for the measurements of the diffusion rate of our EVs and AAV6 in the CF mucus. Interestingly, EVs showed almost 10 to the 0 level. That's the almost 1 micrometer scale level of MSD values. And then EV86 also shows very comparable diffusion rates with EVs. And then in the figures, there is a red dot line as historical value of AV6 diffusion rates. So EVs and EV-AV6 having more than 100 nanometer size, showing some faster diffusion in the CF mucus compared to the AAV6. Usually the small nanoparticle showing some fast diffusion rates. AAV size was 25 nanometer. So it is quite impressive that EVs can penetrate much more faster than smaller AAV6 in the mucus. Then we treat the EV-AV6 to the cells. We use the human bronchial epithelial cell here. This is the simple two-dimensional culture, and we treat the cells with AAV6 and EV-AAV6 and EV plus AAV6, which is the post-mixture. Then only the EV-AAV6 shows much, like, significantly higher transduction efficacy in the 2D culture method. In all multiplicity, in a range of 100 to the 10,000, in all three groups, the transduction efficacy was improved. Then we treat the EV-AV6 to the mouse lungs through the intratracheal administration routes. Then all treated mouse showing some similar coverage of our transgene expressions. But in the EV-AV6 treated lungs, there are much higher intensity of the fluorescent proteins. Then we also prepare the two times for the EV-AV6, but with that two samples, the ratio of EV to AAV was different. Then we just applied that two different batches of EV-AV6 to the lungs. But the low ratio or high ratio of EV-AV6 showing both of them showed better transduction efficacy based on the luciferase activities. And between the low and high ratio, there is almost no differences based on the gene expression. And we harvest the lungs, and then we did the toxicity safety assessments. And then our pathologists evaluate the mononuclear infiltrates and then nuclear fields and then recurses, all values are comparable to the silent. So all AAV6, EVAV6, and mixture treatments recognize it as safe. And then the last part is the airway liquid interface cultures. So we can differentiate the human bronchial epithelial cell, the HBE cell, primary HBE cells, that cells obtained from healthy donor or CF patients. And then we differentiated that cells on the filters with 2 weeks, and then that cells are differentiated. So top side of the cultures can covered with the mucus, and then bottom part is polarized cells are placed. Then we treat our AAV6, EVAV6 samples on the apical side of the filters, and then we analyze the transgene expressions based on the confocal microscopy. Interestingly, in the wild type alley cultures, higher expression of the transgenes was observed in the EVAV6 treated cultures. The post-mixture shows some comparable expressions compared to the AAV6. And also interestingly, CF alley cultures is characterized by the hyper mucus secretions. So there is a much thick and sticky mucus on the top. Even that like harsh conditions, EVAV6 shows some higher transgene expressions, which like AAV6 and EVAV6, EV plus AAV6 didn't show much expressions in that conditions. This is our final data. The conclusion is the EVA6 prepared in HEC cells and then harvested from the cell culture supernatants showed internal and or external associations of AV6 with EVs. And then EVs showed comparable diffusion rate in the mucus obtained from CF patients compared to the EVs. And AV6 association with EVs achieves a great transduction efficacy in a series of evaluations: 2D culture, animal lungs, and then alley cultured primary HB cells. And all treatment groups showed acceptable safety profile, and then association of EVs and AV6 during the productions is essential for the enhancements. The post-mixture separately prepared EV and AV6 shows comparable transduction efficacy to the AAV6 only. This study is conducted with the multiple groups, especially thanks to my PI, Jung, and then our strong collaborator, George Aslandy, University of Minnesota, and then Hopkins, Ken Group, and then Netherlands for the CF mucus and then departments of genetics at HU, and then University of Alabama is thanks to for prepare the alley cultures of the HB cells. Yep. Prepared presentation is over, and then please let me know if you have any questions. Thanks.
Thank you. Great presentation. And I will now allow people to unmute themselves. And while we're waiting for more questions, I will just start. Like, I'm always curious how people decide to work with EVs. Were there any particular paper which, oh my gosh, EVs are so great, let's try to use them as a therapeutics, or it's just like literature search and you see like lots of people use EVs right now as a therapeutic? What was the like a main point why your lab decided to do EV research?
So as we all know, the EVs showing some great delivery carrier like potential. That is the nature molecules, and then many kind of research is focused on the loading of therapeutic agents into the EVs, and then actually after our research is on the literatures based on the EVs and mucus, probably we focused on the loading efficiency about our like plasmid DNA and messenger RNA like that. But I don't know, like there's many questions, but 2012 there is a one paper started with the report EVAV6, not EVAV6, EV associated AAV, and then just one group is continually discover this material. So yeah, so we are like kind of interested to our lab is also work with the AAVs as well. So this is kind of great method to load the AAV itself inside like in the EVs. So we choose these materials and then utilize.
Yeah, just a little bit to what already Gijung mentioned. I mean, it's kind of really natural, you know. It's natural that we use this EV or other groups use this delivery because EV is naturally the communicator between cells, and it's found everywhere in body, which means they should be able to pass through extracellular milling. Just Gijung pointed out in the slide that a few research shows that there's a large amount of EV in the mucus from healthy individuals and then the people with a disease, which means EV can pass through the mucus because they are secreted from the cells and going elsewhere and they're passing the mucus. So that lead to our hypothesis that EV will be a good delivery system to, for therapeutics by inhalation or other mucosal surface. And then as Gijung pointed out, we've been working on AAV6, and then even though AAV is now a clinical product for some few other non-lung disease genetic disorders, but then so far, lung gene therapy has not been successful, and then the main reason is an inability to achieve a therapeutic relevant gene transfer efficacy. So we thought the combination of EV and AAV will synergistically enhance gene transfer efficacy.
Great, thank you so much. And we have a few questions from Philippe asking us. Would you like to mute yourself? And like, since we have like 3 questions from you.
Yeah, can you hear me all right?
Yes, we can hear well.
So I was imagining that you would get to testing this in live animals. So my questions were, first of all, how many AAV per EV? Were there hot EVs and cold EVs? Was there a difference in subpopulation uptake between the epithelial cells you tested in vitro and the mononuclear family alveolar macrophages, et cetera? Compared, and what are the transgenes you kept speaking of? So there was 3 questions. The ratio of uptake, the ratio of uptake of various cells, including the macrophage population, which should be very good at it. And there are alveolar macrophages all over the place here. And then what are the transgenes you're speaking of? Is that understandable, Olesia?
Yeah, of course. I was waiting to see whether Gijung wants to address it, but then it doesn't matter. I can take it over. So I do not remember off the top of my head. The ratio was it varied depending on preparation, but usually most of the time there's more EV compared to AAV. So we believe that it's not because that I think we believe that's because that our preparation simply isolate the exosome population. And then that population includes free EV without AAV and also AAV associated EV. And then when we look into TM image, as you can see, when we found EV AAV, there are multiple AAV. In general, multiple AAV associated EV, which means there are a large quantity of free EV and also some population of the free EV AAV. And then currently, we do not have a method to separate that two populations. So I would say this is a mixture of it. But then based on our series of experiments, we found that the free EV does not affect the gene transfer efficacy of an EV AAV or AAV. So that's kind of inert material in terms of gene transfer efficacy. And it does not also lead to any cytotoxicity. So hopefully that answers the first question. And second question is about the target cells. This method, we do not distinguish the target cells. So it could be any cells in the lung. Based on our cultural study, we know that it transduces many of airway epithelial cells, which include ciliary cells and also, I believe, the goblet cells and perhaps basal cells if they were able to pass through the mucus and also the epithelial layer and get into deeper layer. But that need to be validated in future. Also, based on in vivo study, we found the uniform distribution of gene expression throughout the airway and also alveolar compartment. So it looks like also it transduces alveolar epithelial cells as well. And then perhaps macrophage as well. Even for our non-viral nanoparticles, unless there's a targeting mechanism, they are able to transduce all different type of cells. One important point is, but this EVA, even though they're taken up by macrophage, it doesn't lead to any kind of inflammation based on our historical analysis. But then going back to target cell of a transduction, we will need to further validate by perhaps by flow cytometry and so forth in the future. Regarding transgene here, we tested two different genes. One is a luciferase reporter, both are reporters, and one is a luciferase, just to determine the overall gene expression level. So it doesn't tell us about the where is expression, not even the cellular level in terms of a compartment in the lung, but overall level. Whereas we here also tested the GS green fluorescent protein gene to visualize the distribution of gene expression throughout the lung tissue.
Thank you.
We have one more question in the chat box. Clotilde Therese having a mechanistic question. Would you like to ask the question?
Yes, hi. I cannot turn on the video, but you can hear me, I suppose.
Yeah.
Yeah, I should know, but I don't know actually the life cycle of the AAV, and I was wondering how does AAV ends up in EVs? Is it because the cells undergo death, and then AAV ends up in like dying cell-derived vesicles, or is it, you think, an active process, intracellular process of AAV becoming associated to cellular membranes, either at the plasma membrane or in multivesicular bodies? Any clue, or you don't? I mean, because it could make a difference in terms of whether the EVs are more or less efficient at targeting cells or fusing with cells and delivering their contents.
Yes, that is very critical points about the contents of the EVs during the AAV productions. Our AAV production methods is very typical method for the productions. We transduce the cells with AAV production genes and then incubated that cells for 72 hours. That is definitely safe for the cells. Like after certain time points, more than 3 days, it's like 4 days, 5 days, 6 days, cell can be damaged and then start to dying. So I think this types of EVAV secretion from the cells, we can tell the materials produced in the healthy cells.
Okay, yeah, you think they are not dead cells, it's just live cells.
Yes.
But and do you see the AAV inside the cells before it's released outside with EVs, and is it like located in a particular area of the cell, or is it below the plasma membrane, or that kind of question? Because you did not show an image of that.
Yeah, we did not image at the where the AAVs are produced at the inside of the cells, but there is one mechanism is reported with the recent paper, that is the MAAP proteins can be spliced from the AAV producing genes, and then that proteins is related with the EV secretions, secretion. So AAV secretions also can be co-used at the exerts. So yeah, that is the current clue for now. But it's like that is interested, we can image it them where the association is occurred inside of the cells.
Okay, thank you.
Just to add a couple of points that we do not know exact the mechanism by which EV and the AAV associate inside of cells. I mean, what Gijung mentioned is one of the potential mechanism. Only thing what we know about is it should be some sort of molecular interaction, not like just physical absorption, because when we exert some strong physical force to the collected EV-AAV, like a sonication and then nebulization, they do not dissociate. So we believe there's some sort of a molecular interaction, and then that's something warrants more study. Regarding the inside of cells, usually that's how we and others collect the AAV. We usually the supernatants are removed as a waste, and then after you wash the cells, you lyse the cell and collect the AAV. So during this EV-AAV secretion, of course, there's a large amount of AAV inside the cells. Whether they're as a single, as a state as a virus alone, or associate some sort of membrane, that's yet to be addressed. But then that's hard to answer at this moment, because when we lyse a cell, we kind of disrupt all the membranes and so forth. So there's a possibility that AAVs are just the cytosol and also somehow associated with some sort of membrane, and then after lysis is collected.
Okay, thank you.
One more question in the chat box from Jeff Bufford. Would you like to ask your question?
Yeah, sure. You can hear me all right? Can you hear me all right?
Yeah, we can hear you.
Yeah, okay, sorry. Yeah, I was curious, the speed difference that you had here between these particles was really striking and kind of counterintuitive to me. I mean, is this is a common thing in mucus where larger particles can move through it faster, or do you have any idea what's going on with that? Is it a property of the mucus or the particles, or?
So yeah, that is kind of impressive data we observed, but still there is a kind of lack of understanding the mechanism itself. But we just assume that the nature of the EVs, the membrane, and also the correlated surface proteins can affect us to the diffusion rate in the mucus.
So just to add a little bit on top of it, the mechanism of any particular matter entrapment, a diffusion inside a mucus, is affected by a couple of mechanisms. One is by adhesive interaction. So if any particular matter have any specific or non-specific interaction with the airway mucus, like mucin glycoprotein and the other component, then they'll be trapped. And another mechanism, if anything is larger than pore size, they cannot move, of course, is physical obstruction. And then both AAV and EV, they are unlikely to largely affected by the pore size, because based on our previous assessment and others, the pore size of mucus, even the diseased mucus, is a few hundred, 200-300-ish. So our AAV is about 25 nanometers and EV is about 90-100, and then EV-AAV is 120-ish. So they are all within that size cutoff. So they're unlikely hinder too much by the obstruction. Now it kicks in a adhesive interaction. And then AAV6, we confirmed in previous studies that compared to other EV, they have a better penetration. But then they also have an intrinsic affinity to some glycoproteins and so sugars and so forth. So I believe that this is only speculation at this moment, but then I believe they might be interacting with the mucin glycoprotein to a certain degree, but not to a degree that it completely stops the virus. EV case, there hasn't been any mechanistic studies. So, but then because this is kind of membrane, is negative charge. So, and the mucus is also highly negative charged glycosylated. So maybe there's recursion that kind of facilitate the, that kind of avoid adhesive interaction and facilitate the penetration of EV through the mucus sample.
Cool, thank you.
Thank you so much. We don't have more questions in the chat box. Let me know if you have something. We have a few more minutes left in the meeting. And I have like small question. Do you plan to follow up on the study, do bigger animal research or something?
Sorry?
Do you plan a follow-up on this study? Like to continue explore a mechanism of association and do animal study, larger animals?
Yeah, this is the experiments with the lipoproteins. So definitely we start to work with the therapeutic genes. One of the target is the like cystic fibrosis, CF. And then cystic fibrosis is like highly related with the cystic fibrosis regulation, like transmembrane proteins, CFTR. So we're gonna start to like load the CFTR genes to into the UVAV6 and then treat the disease model.
Great, sounds great. Good luck with this.
Thanks.
And since we don't have any question, we can conclude for today. Thank you everyone for joining us today and hope to see you in the next meetings. Bye now.